I'd like to introduce our third and final speaker for the morning session. I'm pleased to introduce Dr. Norma Andrews from the University of Maryland. Dr. Andrews is currently professor and chair of the Department of Cell Biology and Molecular Genetics at the University of Maryland. She received her PhD from the University of Sao Paulo, Brazil, and she's interested in strategies used by intracellular pathogens to interact with their host cells and the fundamental cell biology of pathogens as they subvert biological processes. Dr. Andrews is a past winner of the Burroughs Wellcome Molecular Parasitology Scholar Award and a Merit Award winner from the NIH. So thank you very much, Dr. Andrews.
Thank you, Beth. Can everybody hear me? So thank you very much to Beth and to Dan for this invitation. It's an honor to be here. And I'm sure I don't have to persuade this audience of how exciting it is to do research at the interface between pathogens and host cells. So this is what we have been doing in my lab for quite some time. And what I have here, just to situate the group of parasites I'm going to be talking about. So they also protozoan, but they belong to this group of trypanosomatids in which you have the trypanosomes, that you actually have two very different groups of trypanosomes. The African trypanosome, that is actually an extracellular parasite. And then I'm going to talk to you about Trypanosoma cruzi, which is called the American trypanosome, which is intracellular. And since we are cell biologists, and what we like to do is to look at mammalian cells and their interactions with parasites, I'm going to be talking mostly about these two groups of intracellular trypanosomatids, so also Leishmania, which you can see here, if you keep this in mind, is how related they are. But you're going to see that there's extremely interesting differences in how these parasites interact with their host cells. So just to introduce, right, the importance of the diseases that these parasites cause. So T. cruzi, which is also called the American trypanosome, maybe it should be called the Latin American trypanosome, because you can see here in red, the incidence of this disease is exclusively in South and Central America. And the reason for this, I'll get back to this, but it's actually related, of course, to climate, but very interestingly also to living conditions, because the cycle, this is a zoonosis in which it's really remarkable. More than 200 vertebrates have been shown to be able to complete the cycle of T. cruzi in nature. So what stops this disease from getting to humans in the United States is exclusively living conditions. So it is present in the south of the US, but there's no active transmission. And this has important implications for the control of the disease. So this, oops, this is not my computer, so I already advanced too much. Let me get back here to what I was talking about. So what we learned over the years with T. cruzi, I'm going to start just telling you mechanisms of invasion. But first, a word about the disease, right? I just mentioned to you that it is transmitted by an insect, but very different from malaria and also from Leishmania that I'm going to talk about later. This is a crawling bug, and this is a bug that hides in the cracks of the walls of these mud huts. So this disease that this insect transmits is through bites. It usually happens in the face area. It's called a kissing bug for this reason. And they are mostly domestic, so they infect usually humans if they have conditions to live in the same places where humans are sleeping. And it's a pretty serious disease, so actually the symptoms, although the acute phase is quite serious, so during the acute phase, which would be this example of this child here, the blood during this stage is crawling with these very motile trypanosome forms that can be detected only during the acute phase, and then the immune response is capable of eliminating a large majority of these parasites. But what happens then decades later, that very serious symptoms develop, and mostly cardiomyopathy is one of the major causes of death of Chagas disease patients, and also very serious enlargement of the esophagus and the colon, which is called the mega syndromes. That also, this can be corrected by surgery, but it's also very serious. So This disease is totally caused in the vertebrate by this very important life cycle stage here, which is the triple mastigote form. And this parasite has a remarkable ability of invading cells. That I'm going to get back into what we surprisingly discovered about this. Once they invade cells, and they differentiate from this very slender, very highly motile form into this rounded form. So these are the intermediate stages that you see inside cells. And they turn into these amastigote forms that replicate inside cells. And for the case of T. cruzi, this happens in the cytoplasm. So they escape into the cytosol. And what you can see, so this is, I knew this was going to happen. So I don't know now how to play a movie with the cursor. Yes, I do. So I'll just show you here an example of what happens right after they have already invaded. They transform into the amastigote forms, and they go back into at the end of something like 4 or 5 days inside cells, they transform back into the triple mastigote stage, which fills completely the cells. So this is what you end up with. So you can imagine this happening in the tissues of the patients. It can happen in many tissues, but they like muscle cells very much, skeletal muscle and heart. And getting to this stage, you can see here that the nucleus of the whole cell has already degenerated, and the cell just becomes this bag of parasites, which they are all infective stages. Each one, when they are released from the cell, which is just happened here, each one of these generates 500 more when they invade another cell. So what we learned several years ago was something very surprising, that we were looking at cells stained for markers for lysosomes, which is LAMP1, this very abundant glycoprotein on lysosomes. And shortly after these parasites invaded cells, so we started seeing things like this, in which the parasite was completely enveloped by lysosomal membranes. You can see in this movie here on the side that this is a live, a cell transfected also with tagged LAMP1. And you can see that the parasite, it's moving, but it's already inside the cell, and the proof of this is that it's completely surrounded by an envelope of lysosomal proteins. So the parasites recruit lysosomal membranes, and this here, what is playing here in the corner, is one of the other important clues that we got for the mechanism happening here, is that this infective stage of T. cruzi, when we perfuse these parasites through cells that have been loaded with a calcium-sensitive dye, you see this explosion of calcium signaling. So calcium is kept at very low concentrations inside cells, as you know, and what happens just by these parasites interacting with these cells is that you get elevation in intracellular calcium, and several members of the lab have dissected these pathways that involve the release of calcium from intracellular stores, and also some calcium entering from the outside, and on the side of the parasites, signaling mechanisms that involve peptidases and generating of agonists that can control this calcium signaling. And we found that the calcium signaling was critical for this process that we call lysosome recruitment. So lysosomes that normally are inside and in deeper regions of the cell are recruited to the site of attachment, and then intracellular parasites are surrounded by vacuoles that have these markers, and then they escape, and the rest of the cycle happens in the cytosol. So over the years, I'm going to show you here more recent data. I think you can see in this movie, which, I thought it was going to play automatically, but probably not. Let me see if this is playing. I'm having to play this from, this is not working. Yeah, that's what I was afraid of, because I'm playing from probably my other movies won't play either, but it doesn't really matter. So this means it's the same thing that is indicated on this diagram here. These are lysosomes loaded with dextran and chased for 2 hours into lysosomes, right? So we were actually able to visualize this process that we had postulated many years earlier, which is this highly unexpected behavior for lysosomes, in which lysosomes were for years, right? In textbooks, they are still considered this end of the endocytic pathway, in which material that enters cells goes through early endosomes, late endosomes, and lysosomes. Then just elevating the cytosolic calcium above 1 micromolar, you can see this immediate fusion of the mature lysosome with the cell surface releasing the contents. And this is what this movie here showed. It was possible to see these puffs of release of luminal content when you just, the only thing that was done here was to stimulate the cell with a calcium ionophore. So this was an interesting concept because it changed a dogma about the behavior of lysosomes. So it is one example of how much we can learn about mammalian cells studying parasites. And this got us into an area that we are still very interested in, which is the mechanism of plasma membrane resealing after cells are wounded. So the history of this is very interesting. It really has been a neglected area in cell biology, but it started in the 50s, in which these groups here working with sea urchin eggs, they noticed by accident when they were just putting cover slips on top of these eggs that the surface of these cells suffered this remodeling, which very extensive blabbing. And if this was done in seawater containing calcium, these eggs could restore their plasma membrane in seconds. And they could even be fertilized and go normally through development. If calcium was removed from the seawater, these cells never resealed and the cytosol just leaked. So this really gave birth to the field of calcium-dependent membrane resealing, in which much later the labs of Paul McNeil and Richard Steinhardt here at Berkeley, they proposed, they did elegant experiments using different mechanisms to wound cells, including laser wounding and microinjection, showing that intracellular vesicles, a population of unknown vesicles, would migrate rapidly to the site of the injury, fuse with the plasma membrane, and then the cell would even increase in surface briefly after this event. So what we reasoned in the lab, since we were seeing lysosomes behave like this, of responding to calcium and fusing with the plasma membrane, we did a series of experiments. Here is just one example of microinjection, and the advantage of using dextran to really track the sites of microinjection is that dextran is very sticky. So you can see here that this red dextran that was in the needle kind of marks where the needle went into the cell. And on these confocal images, you can see that this lysosomal glycoprotein LAMP, a luminal epitope of LAMP, so this proves that it's exposed on the surface, appeared close to the site of the wound. This is one example in which more brute force, in which a razor blade was used to cut the corner of this cell, and you can see also that the lysosomal proteins appear at the site of the injury. So this led over with a work of quite a few people in the lab for quite some time, that during injury, when you have this really strong increase in the localized cytosolic calcium concentration, there is a calcium sensor that we found to be present on lysosomes from this family of synaptotagmins. So the synaptotagmin 7 senses the calcium concentration and facilitates formation of the snare complex that promotes lysosomal exocytosis. So this process is induced at the site of injury. And what we got very interested more recently is what is the role of the lysosomal hydrolases that are being released during this process. So you have represented here, right? So in this case, a pore-forming toxin that is allowing the calcium to go in. This triggers lysosomes to fuse with the plasma membrane. And we have been focusing on one specific, very interesting enzyme, which is the acidic sphingomyelinase. So this enzyme had been shown before by Fred Maxfield, that it's capable, when added to cells on the outside, of inducing formation of endosomes. So what we showed is that this process of endocytosis that follows exocytosis of lysosomes is what appears to be really the event that is removing the lesion from the plasma membrane. So, and the reason that this makes sense is because there has been quite a bit of work lately showing this effect of sphingomyelin, of course, generating the product of this enzyme, which is ceramide, generated from a very abundant lipid in the plasma membrane, sphingomyelin. And ceramide has the property of coalescing in membranes and then of causing membrane bending and invagination. So this is the mechanism that we envision that is happening. And this postdoc in the lab, Cecilia Fernandes, then of course did not miss a beat. And since the whole story started with trypanosomes, I think, again, I may have a problem with movies. No, this one is playing. So she asked the question, do these parasites wound cells when they interact with their host cells, right? So what you can see here, that prior to invasion, these trypanosomagotes, they glide over the host cells for quite some time, and they exert quite a bit of pressure. And you're going to see this because this parasite just moved away, and you can see that it leaves this impression on the membrane. So there's mechanical force being applied there. And using this classical acid that we use for all our membrane repair assays, propidium iodide is membrane impermeant. So if you add it to the outside during an infection experiment, these labeled nuclei here indicate that these cells got wounded by the trypanosomes. So the parasites wound the cells, and what Cecilia went on to show is that if you knock down expression of the acidic sphingomyelinase, the enzyme that is delivered by the lysosomes to the outside of cells, you can see how much it inhibits the entry of these parasites. And she also used bacterial sphingomyelinase, just adding it in a dose-dependent way, she can then rescue this invasion. So the invasion is really modulated by sphingomyelinase. So this is the mechanism that we envision, is that the calcium influx recruits the lysosomes that fuse at the site of the parasite attachment, release this enzyme that immediately promotes bending of the membrane. So for the first time, we're understanding better how this quite large vacuole, that these vacuoles that contain cruzi can be up to 15 microns long. And Cecilia was actually able to visualize this with antibodies against ceramide. So you can see here that this is the staining that we have been doing for years, right? Right after invasion, you see the parasites inside cells surrounded by the lysosomal markers, but she could also see, and in this case it's great because the trypanosomes have this very typical morphology in which even in this sea of ceramide inside cells, it is possible to distinguish that in the vacuole around these recently invaded parasites, there is an accumulation of ceramide. So this is one thing that we learned in addition to the lysosome recruitment, which is an older story, in which it's not a simple recruitment of lysosomal enzymes, membranes, that this parasite is using to enter cells, but there is a local remodeling that probably is the early event that then makes everything else happen. And this involves generation of ceramide. So this is all I'm going to say about the cruzi, and I just wanted to sense the importance of this center here, and we should also discuss the implications for public health, right? So I make a few enemies in the Chagas disease field when I talk about this. This means this is where I come from. I did my PhD with this parasite. And it's true that it's a very serious disease. There's millions of people still infected in the world. The chronic infected population is very large, and this is a big threat for the blood supply, especially in countries like the US who don't screen for this. But the good news is that because of the habit of this vector, the fact that it is a crawling bug, and this can only establish itself on this type of house that has all the cracks, it's quite easy actually to control the transmission of Chagas disease to humans. So with all this research that we have been done, right, I think it's a very interesting topic, the discussion that is going to happen today. What is the role of academia really? So this means we feel a little guilty because we have spent a long time really having a lot of fun working with T. cruzi and learning very interesting things that maybe at the end of the day are going to have more of an implication for understanding how mammalian cells repair their membrane. I'm not sure anything we did is going to help to get rid of Chagas disease because it's happening already. It's great news, and this means we should celebrate great news. This Southern Cone Initiative that was started by the Pan American Health Organization and the World Health Organization has been considered one of the most effective international public health interventions. And it was absolutely simple. I remember as a student in Brazil during the dictatorship, and when a lot of things were very difficult, there were people really saying that it was crazy that there was not better implementation of methods to control transmission, because all you have to do is to spray regularly the homes with insecticide and then monitor for the return of the bugs. Because elimination from the wild is never going to happen. There are just too many hosts. But it is very possible to eliminate. Just a finish to the walls takes care of the problem. And you can see here the dramatic drop in new cases that has happened in all these countries. And there was a goal set when this happened so well that by 2010 it might be eliminated as a disease. And this has not happened because of Central America mostly, which there's still a lot of people living under these conditions, and complex conditions in the Amazon region. So there's very interesting different behavior of the reduvid insects in the Amazon that it may be harder to control because they tend to go back and forth between wild sites and domestic sites. But anyway, this is good news. So since, of course, we want to stay relevant and we want to make a contribution, although we are basic scientists, we have in recent years shifted our attention to leishmania. So leishmania is closely related to the cruzi, but it's a totally different story. So you can see here that there's really a lot of people suffering from this disease, 12 million infected, 300 million estimated to be at risk. And the risk is expanding with development. It's the opposite of Chagas disease. So you can see here that the forms of the disease, the most severe is the visceral form that in certain regions of Africa is very severe, certain regions of India and around the Mediterranean and in South America, particularly Brazil. So different species cause this. Interestingly, the same species that causes the visceral disease in the Mediterranean does it in South America. And then there's a different species in India. But the incidence of this depends, and the different species and the form of the disease is totally related to the species of sandfly. So there's a fascinating relationship between the species of sandfly that transmits this parasite, and you can see here that the parasites replicate in the gut of these insects. And this is very different from malaria, so this is not a transmission through the salivary gland. So the parasites don't migrate to the salivary gland. They are basically regurgitated. They stay in the digestive tract, and they're deposited by the insects when they bite the host. And the other interesting difference from T. cruzi is that they only infect macrophages. So they are parasites of macrophages in the host, and they remain inside vacuoles. That's what made us very happy, that these vacuoles have properties of lysosomes, so we could apply a lot of what we knew about lysosomes to this. But what is really very important to keep in mind is that vacuole control is probably never going to happen very effectively because they are very tiny. You can see here, this is the knuckle of a finger. And you can see the size of these sandflies. So bed nets that can keep this out I usually have to be so thick that nobody in the field wants to use it. So there's a lot of discussion in which the only way really to control leishmaniasis would be either a vaccine that doesn't exist yet. There's something promising beginning to be developed for dogs, which is an important reservoir, but drugs is really a very big need in the field. And just to give an example here for the people in the audience that are not familiar with the forms of the disease, so the different species can cause either cutaneous lesions that can be self-healing, which is the cause of L. major, which has also been getting a lot of press recently because this is also called the Baghdad boil, which a lot of the soldiers are coming back from the Middle East with this type of infection. So it's not the most severe because it is self-healing, but there are other stages. We have been working in the lab with this group here, Amazonensis mexicana, which it's not always self-healing and can cause big complications. And in some cases, it can also cause disinfection of the mucosal membranes that is typical of another species, Brasiliensis. And of course, the real severe that can lead to lethality is for totally unknown reasons that these other species that remain in the skin where they are injected, these species can visceralize. So they can travel to deeper organs, and then they cause, of course, very severe disease. And at the cell biology level, what's very interesting is that there's also differences in the morphology of the vacuole they occupy. If you look at a scanning EM of intracellular mastigote, it's very similar to the morphology of T. cruzi in the cytosol. In this case, as I told you, they stay inside vacuoles. In green here is the lysosomal marker LAMP1. And in red, just the DNA to show where the parasites are. So this group of Parasites mexicana that I'm going to show you the data on, they live in these collective vacuoles, while the other parasites have this really fascinating mechanism that several bacteria have, that each parasite that divides the vacuole with it. Something we know absolutely nothing about either. So just we started these studies because we were working with host responses to infection, so which were macrophage genes that would influence infection. So I'll just give you a little flavor. I don't have time to get into this, but Jude Wilson in the lab, she investigated looking at genes overexpressed in macrophages infected by the parasites, that this expansion of the vacuole that is typical of this species amazonensis is controlled by this gene list, BAGE, which is the gene that really is involved with this diseases that of enlargement of lysosomes. And what was very interesting that she learned is that the larger the vacuole, the more the parasites like it. So this actually expansion of the vacuole is something that this species developed to promote infection. So artificially expanding vacuoles of other species, they grow better. And what Mauro Cortez found in the lab is that he paid attention to this immune ligand CD200, which is a very interesting molecule, which is normally not expressed in macrophages. It's expressed in endothelial cells and in other cells that normally signal macrophages. And what he saw is that infection by just this species, Leishmania amazonensis, enhances the expression of CD200 in cells, induces its expression, while the cutaneous L major form doesn't do it at all. And what he could show is that using knockout mice for this ligand, that the growth of Leishmania amazonensis in macrophages depends on expression of CD200. So this is one more example of so many of subversion of cells in which the macrophage is expressing the ligand to its own receptor to shut down immune responses that can kill the parasite. And what I'm going to spend more time with is to tell you the work of Rim Othman, in which this is a big effort in the lab at the moment, in which we are looking at iron metabolism in infected macrophages. So what Rim found on the whole cell side is that infection with Leishmania amazonensis induces degradation and elimination from the cell surface of a very important transporter, which is called ferroportin, which is the only way in which macrophages have to put iron out of cells. So iron normally is stored with ferritin, right? Iron is a dangerous molecule, cannot be allowed to accumulate. And iron that is loose, it's either pumped out by ferroportin or it is utilized in many reactions in cells, which is called this bioiron pool. So what Rim learned is that Leishmania inhibits expression of this ferroportin. The intracellular iron concentration increases, and this stimulates parasite replication. So something we're very interested in now, that we want to investigate some reports of autophagy induced by Leishmania, because what's missing is how are the parasites inside the vacuoles getting hold of the extra iron accumulating in the cytosol. So turning now to the side of the parasites, right? So the reason that we are very interested in understanding more about the iron metabolism in cells, because it has been for quite some time, right? It's one of the most, the earliest identified, and actually the one that is clearer, the effect, is as a susceptibility factor for Leishmania infections, not only Leishmania, but also Mycobacteria and Salmonella, is mutations in Nramp1. So what Nramp1 does is to pump iron out of late compartments of the endocytic pathway. So iron enters cells with transferrin through receptors, reaches early endosomes, and then this iron is translocated to the cytosol by one set of transporters, and then iron that manages to go deeper is also captured by Nramp1. And this is critical for all these functions that the cell needs to take this iron before it is stored in ferritin, or, like I told you, eliminated from the cell. So pathogens that reside in late endocytic compartments really have a big challenge of getting to this iron before Nramp does. So mutations in NRAM facilitate Leishmania infection. So what have we learned? Nothing was known until 2006, the first publication from our lab, about how the parasites get iron. So here is represented, right, Parasitophorus vacuole inside a macrophage, in which it has been shown that transferrin, although most of it is recycled, can reach these compartments and carry some iron there, which is in the form of iron 3. And what work from our lab showed in recent years is that on the surface of the intracellular forms, there is a reductase that is capable of converting iron 3 and iron 2, which is the only form that can be translocated across membranes. And iron 2 is translocated across the membrane by this transporter LIT1, which is the Leishmania iron transporter 1, that was identified by this very talented postdoc in the lab that actually did his PhD here at Berkeley with Jeremy Turner before coming to my lab, and now he's at the University of Washington. And Chow really completely on his own, this means he had almost all the data for the paper when he first told me what was happening, which is delightful when that happens. He found on the genome, right, that the genomes had just been sequenced, and he started mining it for known transporters, and he found a high identity and similarity with a well-characterized iron transporter in Arabidopsis. So here is indicated the topology on the membrane, and then Chow did quickly with his great training with Jeremy Turner, using yeast strains deficient in iron transport, showing that this gene can rescue yeast, and this is directly on the parasites, showing that once he made a double knockout, that he could rescue this, he could add radioactive iron, and he could see transport on the wild type, not see it on the knockout, and then he could see complementation by adding back the gene. So this transporter has knockouts of this transporter. They are viable in the insect stage, which was great because it was possible to generate it. But once they get into macrophages, you can see here the normal progression. When Leishmania amazonensis infects macrophages, it starts making these giant vacuoles in which the parasites, indicated here in blue, they line up against the membrane of the vacuole, and here is the staining of lysosomal markers, so it's a lysosome-like compartment. And this is what normally happens over 72 hours. But when they are not able to transport iron through LIT1, you can see that some expansion of the vacuoles happens, but then the process is frozen. You don't see replicating parasites, which can be shown here in this quantification. This is increasing number of parasites over time. The knockout mutant doesn't grow, but complementation restores growth. And it's possible also to see in lesions. So the model for this infection in mice is measuring cutaneous lesions on the footpad of mice, and we can show that the two forms of complementation increase the size of the lesions while the knockout is pretty attenuated. So this was quite useful to know, and then I'll show you something that actually we then had to find the reductase because the substrate of this transporter is iron 2, and there was a paper from Mary Wilson at Iowa that she had shown very nicely the activity in the parasites. So another very talented postdoc, Andrew Flannery, he again, mining the Arabidopsis genome, he found a candidate for a reductase, which is indicated here, this is the topology that is predicted to be on the membrane. And like many others of these genes, LIT1 behaves in the same way. When he depletes the medium in iron, the culture medium, there is a strong induction of expression of this protein. The really cool result that this has been extremely useful for us in our subsequent projects is the fact that Andrew showed that he could rescue the growth of these two mutants, right? So this is the knockout of the LIT1 transporter, the ferrosarin transporter, and this is the knockout of the reductase. Both of them were viable as insect stages, but have a defect of intracellular growth in macrophages. But just by flooding the endocytic compartment of these macrophages with a source of iron as cationic ferritin, you can see that it's possible to rescue their growth. So there are other lower affinity methods for acquiring iron, and this really gave us confidence that this is the pathway in which these transporters are working. More recently, what we are really happy with, because we have been lately learning all this, a total new field for us, iron biology, but most of the iron in our bodies is contained not free or stored in ferritin, but as part of heme. So heme is a critical molecule, right, as all of you know, for numerous processes in cells, is essential for the respiratory chain. And again, heme is a very reactive molecule, so it's not allowed to accumulate. So there have to be very important mechanisms to control this. And a very important issue in the case of trypanosomatids is that it has been shown many years ago that they are incapable of synthesizing heme. So they have to acquire it from the environment. So after moving to Maryland, this was a great example of how when you move to the right place, you get to do something you would have never done. So when I had already decided to move to Maryland from Yale, people in the field told me, you have to meet Iqbal Hamza. So Iqbal basically discovered, using C. elegans screens, the first family of heme transporters. So even in mammalian cells, it was not known how cells would acquire heme across the membrane. So Chow was still in the lab. I was lucky. So this time it was more challenging because he did an extremely creative work of sequence gazing, because the identity is very small. But he identified one candidate in the genome that looked more or less in terms of size. He used size as his criteria to look for this, to the transporter that Iqbal had found in C. elegans. And this turned out to be the real thing. And here you can see a tagged form with GFP that goes to the plasma membrane of intracellular parasites growing in macrophages. It also goes to the intracellular compartment using, again, an E strain that is defective in heme uptake. That transfection with this Leishmania transporter promotes iron uptake. And in the parasites themselves, here a spectroscopic detection of an intracellular pool of heme. In this case, we could only make a single knockout, which is also interesting, but it led to a lot of different strategies to try to get the double knockout. Nothing worked. So the single knockout has a reduced level of iron pool, and it is possible to complement this. And the phenotype of this in macrophages, again, is quite strong, that this is, again, growth inside macrophages. The wild-type parasites increase in numbers over time. The single knockout has a replication defect that is quite strong, that you can see here, that you don't see parasites replicating inside these macrophages. The parasites are in red. And very interestingly, Danilo, the postdoc doing this work, he could rescue the growth of these mutants by allowing the macrophages to phagocytose red blood cells. And physiologically, this is extremely interesting because this is something that we learned from Iqbal, which since 90% of the iron in the body is recycled through erythrophagocytosis, macrophages play a crucial role in actually as sources of iron, right? This means ferroportin, this transporter that I already told you, is tightly regulated depending on the demand for iron in the body. So the choice of Leishmania of the macrophage, we find it extremely interesting because it's exactly the hub of iron metabolism in cells. And Iqbal is the one who is defining. There's another transporter, HRG1, that is located on endosomal compartments of macrophages. And very interestingly, it's looking like it is responsible for taking the heme into the cytosol after the red blood cells are degraded. So the parasites are sitting here and probably intercepting this heme before it's pumped out. So we are now working with Iqbal that there's wonderful things that we can do about manipulating these transporters and seeing what is the impact on the growth of these parasites. So just to show you that this single knockout of the heme transporter is avirulent for mice, we cannot detect the lesions on the foot pads of these mice. And here is the assays that were already mentioned by Bill, right? The type of assay in which you grind the tissue, in this case the foot pad, and you look at parasite load, and complementation takes it to an intermediate level, and the wild type level is here. So this is a classical thing in Leishmania. For mysterious reasons, it's very hard to complement to wild type levels. So the message that I would like to leave with you is that, of course, this is another very interesting discussion that I think we're going to hear today. We think that this heme transporter is an interesting candidate for drug development because unlike the other transporters I mentioned, the LIT1 is only in Leishmania, not in other trypanosomatids. But you can see here on the genome of several species of Leishmania, the African trypanosome and T. cruzi, the American trypanosome, there is a very similar, almost identical gene that is syntenic in the chromosome. So if a good drug is developed against this transporter, we think that this might have wide application in tropical medicine because all these parasites cause very serious diseases. So Iqbal is already in his lab screening compounds together with a postdoc in my lab, and we're excited that there are some hits, but very early process. So I'll just go very quickly through this. I just wanted to give you a taste of something new that happened in the lab that was also very gratifying, right? Using new technology now that is possible to do RNA-seq and then look at all the transcripts that respond to iron depletion in the parasites because we knew that some of those genes got induced by lack of iron. So it was really delightful in one shot, although you pay a little, but you get a lot. Those three genes that I mentioned to you that we got the slow way, the LIT1 transporter, the LFR1 reductase, and the heme transporter, they are top hits on the list. And then there's this whole long list of hypothetical proteins that are still not annotated on the genome that we are very interested in knowing now what this is doing because they are responding to lack of iron. And this response of lack of iron turned out to be extremely interesting and again completely unexpected. This very talented research assistant professor in the lab, he noticed, so this LIT1 ferrous transporter is a transporter that normally is not expressed by the parasites. You can see here right after infection, you don't see it. They have to sit there for 24 hours to appear on the surface. They're not expressed on the promastigote insect stages, but Bidioton saw that all he had to do was to remove iron from the medium, and there it was. So it's possible to induce it in promastigotes, and when he did this, he noticed something really interesting, that the iron concentration in the cytosol of these parasites, when he takes the iron out, actually is counterintuitive, but it shoots up. And after we understood what is going on, it's not that counterintuitive because we know the transporter is being expressed. So iron depletion in the medium results in higher intracellular calcium, and you see that it doesn't happen with the knockout strain here. And you can see that their growth is completely different. So this is the normal pattern of replication of the extracellular insect stage in macrophages. And the knockout that doesn't have the transporter seems not to know that there's no iron in this medium, and it keeps going, and then they crash. So this was a big hint that this availability of iron was regulating something important, and I don't have much time, but the bottom line is that totally unexpectedly, Bidiotum discovered that what is happening is that it's regulating transformation into this infective stage. So taking the iron out, they transform, they change morphology, and they express markers of mastigotes, which doesn't happen when you don't have the transporter. And showing here that doing this experiment and using agents that can generate superoxide, he has narrowed it down to a mechanism in which there has been quite a bit of attention lately, that reactive oxygen species, which have been traditionally just viewed as a response from the host against the pathogens, actually it's much broader than that. It's actually a signaling pathway that regulates many differentiation pathways. In cancer, this is getting a lot of attention recently and also in development. So what Bidiotum proposed is that since superoxide dismutases are only in these parasites, they are exclusively iron dismutases. So this means that iron uptake induced by depletion from the medium would allow the superoxide dismutase in the mitochondria to acquire the iron necessary to then generate hydrogen peroxide, which would then be the differentiation signal. So I don't have time to go through the experiments, but he managed to publish this recently, so he does have evidence that this may be what is going on. So I'll just stop here, and I already showed pictures of most of the people that did the work that I mentioned, a long list of people that are not in the lab anymore. These are the current lab members, great collaborators over the years, and the funding agencies. Thank you.
Thank you, Norma. Do we have some questions? Everyone's shy right before lunch. Dan, in the front here.
I'm curious about the Nramp. I haven't followed the field, but I thought it was supposed to be a manganese transporter, but is really iron probably the most important ligand?
Of course, it can also transport manganese, but this means... So it can also transport manganese, but this is clearly iron because people in the field have shown now in many different ways that it's so similar to the transporter that is on early endosomes, right? There was some controversy. The fact that it can transport iron is pretty established. What was controversial for some time is in which direction it pumped. Some people proposed that it might be bringing iron into the phagosome. But that is not what is thought these days. That it probably has the same topology and the same direction of iron transport as the early endosomal transporter, which is actually quite similar. But the reporters for it were built with magnesium. That's probably what you remember, right? It's with Philippe Gros. Yes. There was one more that I'm ignoring. Yes.
I can stop laughing.
We're recording it for the video, so use the microphone.
I'll just follow up on the Nramp question. So I thought you were going to bring it all back and tell us that then, in the absence of Nramp, that the parasites no longer need to acquire iron. So have you done that experiment? If you try to grow the mutants in macrophages that have that non-functional allele of Nramp, do they no longer need to scavenge iron?
Yeah, of course, it's not that black and white. So what it just confers an advantage for the parasites, but they still need their machinery, right? But what we did that was pretty interesting is following the kinetics of expression of this iron transporter that takes 24 hours on an Nramp negative cell. It happens faster on the Nramp wild type. So we could really correlate this with when the parasites are under more stress to get iron. Yeah. Yes?
There's a question on this side. Does anyone else have a microphone?
For the Chagas work, you showed that calcium was really important in the lysosomal fusion. And is that one of the reasons that, say, in heart muscles or skeletal muscles, where there's muscle contraction and higher calcium, and if you depleted calcium there, would the Chagas parasite not go to those cells?
No, absolutely. This means we really think that it's no coincidence, right? This means the cruzi is amazingly non-picky with its host cells. So if you do it in culture, you actually can infect many different cell types. But it is always more efficient in muscle cells. And muscle cells are the ones that have the very efficient machinery of calcium mobilization, right? So in our early studies about this is what led us in the direction of looking at intracellular stores. Intracellular stores of iron are very important. Yes, parasite takes advantage of it, yeah. I think there's one way in the back, I think. Yeah.
Oh, way in the back here.
Are infections, in general, a risk factor for leishmania?
Other infections?
Yes.
That's an interesting question. This means I've been talking to people in the field in Brazil, and they say that, of course, HIV always is, right? So this means there is some, but there's no good studies done, and they're beginning to look at this. So I don't really know of much published data showing that it's exacerbated or if there is a combination with others. I think this is very much in the beginning yet, these kinds of studies. Yeah.
It might be interesting considering that during general infection, iron withholding occurs in the host. It might be a good animal experiment to infect the animal with some infection that's not going to be lethal, and then see if the iron withholding mechanisms augment leishmania.
Mm-hmm. No, absolutely. I know what you're getting at, because exactly, I didn't have time to get into this, but a master regulator of ferroportin that we know influences leishmania infections very strongly is hepcidin, which is this peptide made in the liver. And one of the main situations in which hepcidin is produced is during bacterial infections. So inflammation signals trigger a peptide that induces degradation of ferroportin. So the ironic thing is that we are interested in following up on this. So one hypothesis is this, the function of hepcidin in suppressing iron transport into the bloodstream would be a mechanism of defense against extracellular pathogens that need the iron. So if there's less iron leaving macrophages, it's bad for the pathogen. But in the case of leishmania, we just learned that it's the opposite. They are inside the macrophage, and if the iron stays there. So we are very interested in this angle, that it may not respond in the same way as it does for bacterial infections. It could be that actually leishmania gets an advantage from the mechanism that is being used to fight infections, yeah, which that is much better understood.
I need to steal your microphone because this one's not working right. Sorry. So thank you everyone for your attention this morning, and let's thank all of our speakers one more time before we go to lunch. We'll reconvene back in here at 1:10 p.m. for the panel discussion. I would just ask that if you see panelists or speakers from the afternoon, to let them get their lunches before everyone makes a mad dash to the table. But lunch will be provided in the atrium. So thank you very much.